Alright everyone, welcome back to this year's Valentine's special! I'm Ink, a microbiology student and VTuber, and today we'll be talking about bacteria sex. No, seriously. Bacteria sex. Okay, it's actually called conjugation and horizontal gene transfer, but functionally it's pretty similar to how we humans reproduce. Kind of. As bacteria are tiny single-celled organisms, everything they use to live is controlled by one gigantic circular chromosome with certain exceptions. You see, bacteria can have these little mini chromosomes called plasmids floating in their cytoplasm, which can contain genes for things such as antibiotic resistance. In order to spread these genes around, the plasmids have to contain the instructions to construct something called a sex pilus. This is a hollow tube made of proteins, designed especially to transport single-stranded DNA from one bacterium to another. Think of it like spending a night passionately hand-holding with your significant other and walking out the door knowing the answers to next week's exam. Anyway, the decision to conjugate is made through the combination of several factors, such as what phase of the growth cycle it's in, or even what its neighbors are doing. Once the decision has been made, the bacteria starts activating the Tra genes one after the other, which tells the cell to start assembling the sex pilus. Now, the bacteria goes ahead and extends that thing into the unknown, hoping to find an eligible partner to exchange genetic material with. Unfortunately for these guys, gay sex does not exist, because bacteria have genders, F-positive and F-negative. If the plasmid being transferred is an F-type plasmid, and the recipient also has the same thing, the two physically cannot connect. But, if the other bacterium is F-negative, the two can connect just fine and initiate the gene transfer. Now, a special mention for those F-type plasmids. These guys also contain a fun little toxin and antitoxin system, which causes bacteria to literally be dependent on it to live. The toxin is very stable and sticks around for a long time, and it's constantly made in the cell at a steady rate. However, the antitoxin is far more volatile and gets degraded rapidly. In order to live, the bacteria has to constantly make antitoxin so that it doesn't, well, die. Now, if by some chance the bacteria does the conjugation tango and loses its copy of the F plasmid, well, it fucking dies. Going back to our analogy from earlier, it's like if you someday forgot the answers to that exam, you'll suddenly die to, like, food poisoning or something. Now, isn't that lovely? Anyway, I hope you guys learned something new today, even if it's a topic nobody asked for. As always, references and further reading can be found in the description. Thanks for listening, and I'll see you guys next year.